What's up, guys? I want to share a little tip with you. When I was studying leukocyte adhesion deficiency, the subject of neutrophil margination came up. And that's the whole process, which I've drawn out a little diagram right here. This is the process where a neutrophil is going through the blood and eventually leaves the blood and goes into the tissues to fight infection. And that's basically the simple way to explain this. And we've all learned that the first step is going to be vasodilation, where this vessel will kind of bulge out at the postcapillary venule, and that pushes the neutrophil to the edges here. And then the second phase is going to be rolling, where it slows down. And we know that in the process of rolling, that involves Sialo-Lewis X. And then we know Sialo-Lewis X and the selectins. And then we know the process of adhesion. And adhesion involves the integrins with the ICAM1 and then the diapedesis, which is actually the actual crossing. I'll just put DIAP. That's the actual crossing over. So I want to share a tip with you in this. I want to share a tip. Let me clear up some space now that I've written a little bit. I want to share a cool way that I remember basically what happens in the process of margination. How do we keep track? Say this is a neutrophil and this is an endothelial cell. How do I figure out what specific molecules are on the neutrophil and what specific molecules are on the endothelial cell of those four things we named? For example, in step 2, I said that this was rolling. And I said in rolling that Sialo-Lewis X, involving Sialo-Lewis X binding with the EMP selectins, right? But I've seen test questions where you need to know, okay, it'll ask you specifically which one is Sialo-Lewis X on and which one is EMP selectins on. And then same thing for step 3. When you move into the tight adhesion, the tight binding, you know that involves integrins, right? Integrins, another name, you could say CD18. And then it also involves going to ICAM1. How do you remember where all of these different things go? You may mix it up and say that neutrophils, you may put something wrong with the neutrophils in the endothelial cell. So I'm going to show you a tip of how to keep all this straight, because this is kind of important in leukocyte adhesion deficiency, because we know, leukocyte adhesion deficiency 1 isn't that complicated to understand. The symptoms are pretty diagnostic. Delayed umbilical cord falling off, right? The reason that happens is because the neutrophils can't get to the site near the umbilical stump, and they can't begin the process of necrosis because they can't get through the wall, right? So that's the first main one. You're going to have, in this process, you're going to have an increased neutrophil count in the blood. That's because the marginating pool that's in the lungs, which normally holds the neutrophils onto the lung wall, this process that holds them there is using adhesion, and adhesion isn't working properly. So it's basically letting go of all those neutrophils in the marginating pool, and now they're all floating around in the body. So you're going to have increased neutrophil count. We know you're going to have tons of infections, so recurrent infections, right? Tons of infections, specifically a lot of mucosal ones. The reason they're mucosa is basically at the entrance of the body where you're prone to take in a lot of pathogens and stuff, and the neutrophils can't not get to the site of the mucosa because they can't pass through the endothelial cell into the tissue site there. And so that's just some of the main symptoms. That's not all of them, but that's kind of how you can recognize it. But the point of this video is to keep straight. LAD1 is involving a mutated CD18, or you could say integrin, right? Mutated integrins. Okay, the question you may know that information, but the question could put that the integrin is on the endothelial cell or the integrin is on the neutrophil or any of these other segments. You could say, where's the Sialo-Lewis X go and whatnot. So this is a tip to help you keep it all straight. First one, Sialo-Lewis. Sialo-Lewis is involved with the rolling phase of neutrophil margination. Well, that's the phase that slows it down. So how do we know if Sialo-Lewis is on the neutrophil or the endothelial cell? Well, here's the way I do it. I deliberately pronounce Sialo-Lewis as sialoleukocyte. Sialoleukocyte. And we know that neutrophil is a leukocyte and endothelial cell isn't. So we know that Sialo-Lewis must be on Sialo-Lewis. We know that Sialo-Lewis must then therefore be on the neutrophil. So that's the first one. So the next one, E and P selectins. This one's kind of easy because if you can remember that E and P part that goes in the beginning of it, E stands for endothelial. So that's the answer right there. E stands for endothelial. And P stands for platelets. Now, that's something you would think, well, why is a platelet selectin in the endothelial cell? Well, the platelet selectins are exclusive. Like if within platelets, that's the only selectin you'll see. But these are also found in endothelial cells. So if you can remember E and P, you know endothelial cell, endothelium and platelets is all involving the endothelial cell. So that would then go here, the endothelial and P selectin, right? So, okay, so we've figured out now the rolling phase. But the LAD1 deficiency takes place in the adhesion phase, specifically with CD18, the integrins, right? So how do I figure out whether CD18 goes in the neutrophil or the endothelial cell? Well, pronounce integrins as integrins, integrins. It sounds like that, integrins, integrins, see? So that way, the N stands for neutrophil, integrins. And then you'll say, well, what about CD18? How do we remember about CD18? CD18, listen how it's pronounced. 8. Like somebody ate something. 18. And remember that a neutrophil is a type of phagocyte, right? It's a phagocytic cell. And phagocytes, they basically engulf or they eat the pathogens that are invading the tissue. Besides just neutrophils, macrophages are also phagocytes. So if you can pronounce it CD8, like you ate something, that has to be on the neutrophil again. So there you go, integrins and CD18. And then, well, then if you know that the other one, CAM, must go onto the endothelial cell. So that's just a short little video. This is a tip that helped me a lot because I was constantly missing questions on a process that I understood but couldn't remember which of these different substances or different units were on which, whether it was on the neutrophil or the endothelial cell. So I hope this helps a lot. Please like, subscribe, and share the video if it helped you in any sort of way, and I will see you in another video.